Hello and welcome everyone to another episode of Biology with Arseni. Today we are going to talk about enzyme, why enzymes are important, what they do, and what we do make of it. So we're gonna know after the intro. So as I was saying, today we're going to talk about enzyme and why they are important and why do we need enzyme in the first place. Well, in the first hand, we should know that enzymes are proteins and they are made of amino acid, of course. And since we talk about proteins, we should know that they are protein in their tertiary structure. Okay, and that's make them function properly and know what they are going to do um, from the chemical reaction. Enzymes are called a biological catalyst. They do accelerate the rate of the chemical reaction by lowering the activation energy needed. Every reaction within our body needs a certain amount of energy to start. That energy, well, it's it need a big energy. It need a higher energy outside our body, but inside our body, it is manageable through enzyme because enzymes enable these reactions to happen by lowering this activation energy. Okay, you will see at your right that there is a diagram shows you the difference between the energy needed without the enzyme and the time taken and with the enzyme and the time taken for it. You will see that there is a huge difference between them. The blue line is without the enzyme and the red line is with the enzyme. You'll see significantly that there is a difference between both. Now, this difference in activation energy, it enables these reactions to happen within our body smoothly without any kind of problem. Without the enzymes, well, we're gonna need a higher amount of energy, which apparently isn't available in our bodies, and it will make a huge problem to our body on the other hand. So there is clearly a difference between something that we call it enzyme and their cousins, the ribozymes. Now the enzymes they are made from protein and the ribozymes they are made of RNA. To be specific, they are made from ribosomal RNA. Both of them they do somehow kind of functions similar to each other, but they are different in their composition itself. Now, the enzyme, they are separated into two groups. Either they are simple enzymes, which they are made of protein only, or they are conjugated enzymes, which they are protein enzyme and some other things with it. Okay, made of protein part, which we call it the enzymes, or non-protein part, which we call it a cofactor. Both of them, we call them the holoenzyme. So the conjugated enzyme, okay, we can call them a holoenzyme, which made of the previous mentioned part, the enzyme and the cofactor. And the cofactor, they can be organic, it can be a thermolapile, and they can be loosely bonded, and they are mainly made of vitamin B and its derivatives. Okay, that shows us why vitamin B are one of the most important vitamins within the human body and why they are so needed in a fairly nice amounts, enable our body to perform and our cell to perform properly because they are needed within the enzymatic system, and that enzymatic system clearly helps what happened within our cells. Without this enzymatic system, well, you'll have a huge problem within your cells, and it's gonna make it not nicely happen. In order to understand how the enzyme work, we should know the mechanism. That makes me wanna assure that enzymes, the main goal of them is to speed up the reaction without being changed. I mean, they go in, go out, unchanged. They aren't participating in any kind of reaction or whatsoever. They are simply doing a very simple job. They're going to speed up the reaction by lowering that activation energy, and that's it. They don't need anything else. Okay? And the first mechanism we should know of is the lock and key mechanism. In the lock and key mechanism, we have a place within the enzyme we call the active site. Okay, it is in a complete fit with the substance that we call the substrate that works on it. The problem here with the lock and key mechanism, before we talk about it, is it assumes that the enzyme has a geometrically stable and geometrically fixed shape. And that is not the case for the enzyme. Assuming something like this, that means that I'm going to assume that all the people around the globe, they may be in a circle shape, they can be in a rectangle shape, or a square, or a triangle, or a parallelogram, or whatsoever. Okay, but that is not the case. That's what does not happen. Okay, the idea of the lock and key is to enables us and help us to understand how the enzyme just how it works its magic, and how it help in separating things and make everything goes away and nice. Okay, so it starts with a very simple step. We have the enzymes with the active site in place. Okay, and the active site, it has a complementary shape to the substrate, and the substrate with its shape come to this fixed place. They fix together, and like the locking key, for every lock there is a specific key. After the enzyme molecule work their way and they help the reaction to goes on and on, the results goes away and the enzyme stays at its place. Normal shape can be reused hundreds of times. Okay, the enzyme will continue work as long as there are substrate that needed to be worked on. Okay, so that's the lock and key mechanism. So the pros, or the things that we say, that the positives of the lock and key theory, that helps you to get some kind of knowledge and some kind of insight what happened, what the enzyme do, and how the enzyme work. But the cons on the other side, well, it is fairly simple. It assumes that the active site is geometrically stable, which isn't the case. So everything has its pros and cons. So there was a need to have a better understanding how the enzyme work, and here came the induced fit model. In the induced fit model, the active site isn't completely fitting with the substrate. Well, that means that the enzyme can go through around the body or around the cell without any kind of bonding, or without any kind of enhancing any chemical reaction or whatsoever, until it finds its specific substrate. At that point, the active site starts to bend over and change its shape to accommodate the substrate that it works on it, to fit the substrate. After that, the enzyme work its magic and work its way, completing the chemical reaction and move to another one. So here, the active site isn't ready to receive that substrate. On the second step, now the enzyme substrate complex, the enzyme starts to change its shape to accommodate the substrate. The enzyme product starts to come, and the product moved away. Okay, leaving the active site back again to its normal shape, where it isn't compatible to any other kind of substrate or whatsoever. So that is how the enzyme works. But there are several factors that affect the enzyme activity. We're going to talk about 4 of them today. I'm going to leave the other things to another time later down the road, because that's not the enzyme first time we're going to talk about it. We are going to talk about the enzyme several times down the road. So the first factor that out of around 7 or 8 factors affect the enzyme activity is the enzyme concentration. The second one is the substrate concentration, temperature, and pH. Okay, there are also the time, the coenzyme if they are present, or if there are any kind of inhibitor that stop the enzyme activity from working. So I'm going to talk firstly about the enzyme concentration. Now, there is a direct relationship between the action of the enzyme or the rate of the enzyme action and the enzyme concentration. Well, as long as you give a high concentration of enzyme, you will get a faster reaction because now you have a better surface area, you have a larger area where you can work its way around the substrate and you can finish the chemical reaction as fast as you can. So we have a directly proportional relationship between the enzyme and its speed. On the other hand, the substrate concentration itself. So you have a huge amount of enzyme. Now, if you have a huge amount of substrate, the enzyme will start to work its way fast until it will reach, let's say, a maximum amount of product and the reaction start to stabilize. And we can see it here in that graph. You'll see that the graph over here, it goes all the way up very fast, very steep until it reaches a certain point and the reaction goes flat. Going flat, that means that you cannot get any higher results from that reaction. It simply shows you now I reach the maximum capacity of where I can work and where I can perform properly. So these are very two important factors that affect the enzyme activity. But there are another two most important factors that affect the enzyme activity, which is one of them is the temperature. Each enzyme has an optimum temperature where it performs efficiently at that temperature. Okay, now as you can see here in the graph, you will find that there is an optimum temperature for each enzyme. The enzyme activity starts to work its way and it starts to grow and grow and grow until it reached the peak over here. If the temperature had exceeded the what the enzyme can take, now the enzyme concentration starts to fall down until the enzyme itself disintegrate. Okay, and by being disintegrate, now the enzyme, as we said, it is a protein, as we mentioned in the protein episode. I'm going to leave it link in the description above and in the link in the description below. You can check it. And since you have reached this point, make sure to subscribe, turn on the bell button so you can have the latest update from our episodes from Biology with Arseni. I'll be thankful if you subscribe. It will be great for me, and it will help us to spread the science much more. So what happened to the enzyme? It started to twist, be bent out of shape, start to disintegrate. It is not in its normal shape, so it will not work properly. But if the enzyme starts to go back down, so it is like freezing the enzyme. So freezing the enzyme, of course, it will decrease the stability of the enzyme. It will decrease the activity of the enzyme, but it will not disintegrate the enzyme. It's like keeping things in the fridge. You can get it out, you can deal with it later when it's thawed out. So after the optimum temperature, well, your enzyme is done. You can't do anything with it. But if it's with the temperature below, well, you'll have a decreased efficiency. If you continue to drop the temperature, you will freeze it. You can reuse it again, but the enzyme always prefers to be in optimum temperature. The enzymes in our body, well, its optimum temperature is around 37 degrees Celsius, maximum 37.5 degrees Celsius. That is the optimum, and that is the best temperature where the enzyme. The other part that affects the enzyme activity is the pH, and the pH is the concentration of the hydrogen. Now, In order to understand the concentration of the hydrogen, we got to know that there is something that we call the pH meter. The pH meter ranges from 0 till 14. Now 7 is the neutral number, below 7 till we reach 0 is the acidic, above 7 till we reach 14 is the basic, or some people may call it alcohol. Okay, so an example for this, we have the pepsin enzyme within our stomach. It works in acidic pH of a pH 2 to 3, or we have the amylase enzyme within our mouth. It's helps in degrading down the sugars, and the carbohydrate, it starts to work around 7.5 till 7.7 pH. The trypsin enzyme, not the trypsic, the trypsin enzyme, that's a spelling mistake from me. The trypsin enzyme, it works at the basic pH around 8 pH. Okay. Now for also every enzyme has its optimal pH. Some enzymes work in a basic. Some enzymes, and I'm going to show you here, it starts to work in acidic. I'm not going to draw it in pink. It will be the same. I'm going to draw it in green. It will be much brighter, and I like green. So this is it. It starts to grow. That's the optimum. Let's say the optimum is around 3, and it falls down near 6. Okay, so that's the optimal temperature. So we can give it to the pink one over here. We can say this is the trypsin. Okay, and the green one is the pepsin. Okay, now these are several factors that affect the enzyme activity. This is how the enzyme works. Again, these enzymes simply makes our cells to perform their chemical reaction much more easier way. It helps the cells to work their way in a proper way and in much easier way. So that is going to be all today. Thank you so much for watching. Don't forget, like, subscribe, share all around the social media so everyone can get the benefit. Until I see you next time, I was Arsene Aziz, and that was Biology with Arsene. See you later.